The concept of omics sciences unites disciplines such as genomics, proteomics, and metabolomics. Each branch contributes significant advances to the basic understanding of biological investigation. Furthermore, they represent enormous development in the field of cell function analysis and its biotechnological applications. Scientists from the Príncipe Felipe Center for Investigation develop different research programs for discovering new markers for diagnosis, prevention, and new treatments that help doctors individualize prescriptions for each patient.
Our main goal is to try to use the genomic data. We are in the genomic era, so our idea is to develop methods, develop algorithms, and to implement them in programs in a way that can be directly used for the clinicians. We belong to the CIPF of rare diseases. The CIPF is the Spanish network for the study in rare diseases, and we also are very focused in cancer.
Some of these translational applications allow for the sequencing of the patient via his or her genomic information. In this way, one can identify variants of the genome that are associated with an accurate diagnosis and adequate treatment. All of the processed information is stored in a database, giving way to a process of permanent feedback.
In that way, we can implement easily the personalized medicine in the clinical system.
One other of the developed applications has the objective of transforming the results of measurements of mutations or genomic activity with cellular function in order to directly relate them with phenotype. This is done through the use of graphs that show cellular functions. We work with graphs of interactome function and regulation, and particularly those of signaling and metabolic pathways. By superimposing one graph over another, one can observe which functions are more important and which are irrelevant. Investigations of the genetic and epigenetic expression program has two main goals that are tightly interconnected.
On one hand, we are collaborating a lot with clinicians and medical doctors at hospitals and at research institutions, and they have the clinical problem, and normally they will also have the patients, they will have the essays. So what we do is we collaborate with them, they provide us with the data of these patients, and then it's our role to analyze this data and to transform the data into knowledge. The other source of information is the data which is available in public databases. This big information is stored in specific places, and these places are accessible to the scientific community. So we can take this information and then connect this information with the data that is coming from particular patients, and that gives a lot of power to the statistical analysis of the data. We are one of the first groups that have started to combine very many different types of genomics data, gene expression data, epigenomics data, data related to the metabolism. So what we try to do in the combination of all this data is really understand the functioning of biology from all the different aspects that a cell is working. So we have been pioneers in doing this integration.
Software designs such as Stratega, Blast2Go, or Team are some of the translational tools developed by experts that allow one to directly connect investigation to clinical medicine.